Yes, sir.
Okay, all right. Well, welcome everybody for joining us tonight. For those of you that have not met me, I'm Sean Kramer. I'm the CEO for the Diabetes Research Institute Foundation. And I myself have been impacted by diabetes. If you don't know my story, I'm a fourth generation person in my family living with diabetes. I was diagnosed now about 6.5 years ago. And I am a patient at the DRI. And I came on board as the CEO for the DRIF now in January of 2020. So right before the pandemic started. And that was really the impetus for us to start these Meet the Scientist events. So we're excited that you're here joining us for this special opportunity to meet with the DRI scientist, Dr. Baidal, which I'll introduce in just a moment. And we hope that you'll find the session really to be informative and hopefully be inspired by really the critical work and amazing work being done right now to cure diabetes. For those of you that aren't familiar with the DRI as a whole, I know many of you are that are on the call, so we appreciate that. We lead the world in cure-focused diabetes research. We're one of the largest and most comprehensive research centers dedicated to curing diabetes. And the DRI is aggressively working to develop a biological cure for diabetes by restoring natural insulin production and normalizing blood sugar levels without imposing other risks. And the mission of the DRI Foundation is to provide the Diabetes Research Institute with the funding necessary that they need to cure diabetes now. So we thank you for the critical role that you all play in curing this disease. And the groundbreaking work at the DRI and beyond would not be possible without supporters like yourselves. So thank you. Just a brief overview of the session. Dr. Baidal is going to talk for about 20 to 25 minutes, and then we will do a Q&A. So hold your questions until then. You can always type them into the chat so we can get to them in case something comes up that you want us to circle back on. Please go ahead and do that. And with that being said, let me introduce our wonderful guests for the evening, Dr. David Baidal. So David is an assistant professor of medicine in the Division of Endocrinology, Diabetes, and Metabolism at the University of Miami Miller School of Medicine. And he's also a member of the DRI's clinical eyelid transplant program. He received his medical degree from the Universidad Católica Santiago de Guayaquil in Ecuador. He completed his internal medicine residency at Jackson Memorial Hospital, which is part of the University of Miami, and then went on to complete a clinical and research fellowship in endocrinology, diabetes, and metabolism at the Beth Israel Deaconess Medical Center, Joslin Diabetes Center in Boston. So with that being said, I'm going to turn the program and podium over to Dr. Baidal.
Thank you for the introduction, Sean. I'm very happy to be here and for the opportunity. Just wanted to take advantage to give a little bit of additional background of my ties to the DRI, which I've been involved with the DRI for over more than a decade. And it's interesting, when I was doing my medical training in Ecuador, I became very passionate about diabetes because my grandparents developed type 2 diabetes. So initially my focus, I knew right then during my training as a physician that I wanted to work in the diabetes arena, but with type 2. And I had the opportunity when I was about to finish my training to do an observership through a Latin American program that was available at the University of Miami, at which time the Diabetes Research Institute was actively participating, allowing foreign medical graduates or foreign students to come and rotate through various specialties in the University of Miami. And I found in the list of rotations, one that said diabetes research, which immediately caught my attention. And I was very fortunate that that rotation entailed a rotation with the clinical cell transplant program with Dr. Alejandra and Dr. Ricordi. It was back in 1999. And you can imagine for somebody that is just learning medicine, coming here and being exposed to, it was actually the right timing when eyelid transplantation was having this peak of success. I was in awe and I fell in love with the research of the DRI, and I shifted my interest from type 2 diabetes actually to type 1 diabetes. And what I initially thought would be a 2-year relationship with the DRI, where I will get some information and some training in clinical research and then go on to do my residency training. It translated into actually 9 years, where I got a huge amount of training in research, obtaining a position of an assistant scientist, and ultimately going to do my residency in Jackson here in Miami, and then do endocrinology in Boston, and ultimately coming full circle as an endocrinologist back to the DRI, working again with Dr. Alejandro and Dr. Ricordi with the mission of this institute that I hold very close to my heart because it has had a huge impact in my training over so many different years. So with that being said, let me just quickly share my screen and my slides because I would like to show you, I think I'm not sharing the correct screen, bear with me for 1 second, a little bit of what I do so that you can see what wakes me up every day and the excitement to continue doing what I'm doing here at the DRI. So hopefully you can see my slides. And I really like this that was done by the foundation. I appreciate it. It looks very nice. It provides a little bit of the background. And let me start off. I think that since I'm going to be talking about what I do as a clinical scientist with the clinical scientist hat, because I also do see patients and I'm an adult endocrinologist with a focus clearly in type 1 diabetes. But when I talk about the research, right, I think we need to go back to the basics of understanding a little bit of the pathogenesis of the disease. As an institute focusing, and I'm hoping you can see my mouse. I hope you can. Let me see if I actually do this, put here the laser pointer. Maybe that will be better. Clearly, our institute is highly renowned, and we have moved the field with the use of a cellular therapy, which are islets. But we use that to treat patients that have established disease. So way down the natural history and the pathogenesis of type 1 diabetes, right? So here is where we're intervening to provide a cure so that those patients that are affected with long-standing type 1 diabetes can ultimately have restoration of their insulin production. We replace the cells that have been lost and with the intent of curing them. But clearly the disease starts much earlier. And if we understand that there are different stages, this opens a window of opportunity to intervene earlier. And I think the uniqueness of the DRI is that we have the opportunity to collaborate with other centers and other scientists, and also with some pharmaceutical trials, so that we can tackle the disease not only down here, which will benefit a lot of patients clearly, but also earlier to try to prevent the disease from happening or to slow down the loss of insulin early on in the disease process. So we divide diabetes in different stages. Pre-stage 1 being those relatives, those patients or those kids that have family members that have diabetes. So they have a genetic predisposition. But when they're born, they have all the functioning beta cells. So if we look here at the line here, imagine this being 100% of your beta cells. But you have the genetic predisposition. Then something happens that triggers the immune system. We don't know what is the trigger, unfortunately. But we do have something called immune activation. Your immune system is now active, and we have a way of identifying that because now you actually develop antibodies in the blood. And you can test for those antibodies. And if you find 2 or more antibodies, you are at risk. Your risk of developing type 1 diabetes actually increases. And it is at that time point, when you develop the antibodies, 2 or more, that you start losing your beta cell mass. Your insulin-producing cells start to come down. However, you still have normal sugars. You still have normal glucose tolerance. So if you just monitor the blood sugars, they will be normal at this stage. As the disease advances, sugars become abnormal. And then you enter what is called stage 2. So now you have the antibodies and the blood sugars are now abnormal, but you don't have diabetes yet. So it's like a pre-diabetes type of stage. And ultimately, diabetes gets diagnosed in stage 3. Now you have the symptoms. And down the line, years later, you lose more of that beta cell mass and you have established disease. And each of these stages provides an opportunity and a window of treatment. And I have been fortunate to be involved with clinical trials in all of these stages. When we go, again, back looking at this this cartoon, I have been fortunate to be involved with multiple trials of islet transplantation in established disease. As you can see here, looking at clinical trials of cellular therapies, islet transplantation alone, patients that have received bone marrow with islets, islet after kidney, islets with different drugs. We played a critical role in a phase 3 trial of islet transplantation showing efficacy of this treatment. And we have also been pioneers in showing that a different site, islets, are typically transplanted in the liver, which is the site that has shown success and it works, and I'll discuss this a little bit further along in this presentation. But we know that the liver is perhaps not the ideal place. So we've been looking at the omentum as well. And this is a trial that is ongoing, and we'll show you, again, a little bit of data regarding that particular trial. In addition, there's another trial that we're recruiting for, which will test transplanting islets in the eye of patients who are legally blind. And most recently, we have been fortunate to participate in a clinical trial using embryonic stem-derived islets. And more of that to come later in the presentation. And when we shift from cellular therapies and go into using immune therapies to try to modulate the immune system and decrease inflammation, I'm happy to be the principal investigator for our POSEIDON study, testing omega-3 fish oil and vitamin D, which is ongoing. But now let's go further back in terms of clinical trials. You can imagine that in patients that have just the generic risk, they don't have the disease yet, and they don't have the trigger yet. We do have monitoring studies to try to determine what could be potential triggers. For patients who are at moderate risk, those that already have the antibodies, so you have immune activation, I have been involved through partnership with TrialNet with monitoring studies, the use of oral insulin, and there are two other trials that are ongoing that I'm participating with, which is the use of these two drugs, abatacept and hydroxychloroquine, all of them with the hope of preventing disease. In patients at high risk, these are patients that, again, have antibodies and their sugars are already abnormal, but yet do not have diabetes. So they have, it's kind of like a pre-diabetes stage. There is a trial that tested a compound, an anti-CD3 agent called the Plysma that I will show you shortly, that actually showed efficacy. And this is unique, and I will talk to you a little bit about that in a minute. And then it's here in the new onset realm that we have been involved in multiple trials. I have not been involved in all of them. I have been involved in a few of them. And the reason why there's a little bit of a gap and a difference in clinical trials early on as the first two in patients who are newly diagnosed, it kind of like makes sense. It's difficult to justify medications, immune therapies that have side effects in a patient that doesn't have diabetes yet, right? And we typically restrict it to adults. It's even more of a challenge to justify therapies in children without the disease. It's easier to justify it in somebody who has recently been diagnosed. And we have data from animal models and studies showing that if you intervene early after diagnosis, you're more likely to succeed when you implement immune therapies. And again, we've been very fortunate to participate in several trials, and we have learned what doesn't work, and we have also learned a little bit of what therapies actually may work in new onset disease. So now let's go back to islet transplantation, which we have been leaders in the field and really have moved the field forward. What has been our experience in Miami? So looking at 56 transplant patients since 2000, on the left panel, you're going to look at graft function, meaning these are patients that get a transplant and the transplant still makes insulin. We can see that up to 20 years, over 50% of those patients, still those transplants are making insulin. And if you look at insulin independence on the other panel here on the left, depicted in red, by 20 years, you only have about 10%. So insulin dependence is lost. But the purpose of this therapy, because again, we're evolving, we're learning, we're trying to optimize it, but the purpose is to actually improve glucose control, decrease the variability, eliminate severe hypoglycemia. And in those patients that maintain function of the islets, their own insulin production, even though they're not insulin independent and they're using much less insulin than before having a transplant, we have been able to achieve almost normal A1Cs, and that means an A1C of 5.6 or below. So we're not talking about less than 6.5, we're talking values of less than 5.7, without severe hypoglycemia, and an improvement in the quality of life. That's the impact that islet transplantation can have. So it should not be viewed from the perspective that I'm not off insulin. What is the metabolic control and the quality of life gain that you can obtain with this therapy. And the question is, what about those that actually are able to remain insulin independent? Are they truly stable, the glucose? Because it's easy for us to say, just stop your insulin. Are your numbers really where they should be? And this is a figure that shows data which you may be familiar. These are continuous glucose monitoring data technology, showing 5. So each one of these lines represents an individual subject. And here on the left, you have the blood glucose, the sensor glucose data. And the edges here, these 2 lines are the 70 to 180 range. That's the target that you want to achieve for management of diabetes. And you can see that most of the time, these patients are able to stay. This is over a 24-hour period, and it is derived from 1 week of data. They're able to stay in that range. So these are, all of them are off insulin. It's an average of about 11 years after being transplanted. And when you look at the timing range of 70 to 180, 96%, with almost no time spent having low blood sugars. And the coefficient of variation, which is a metric that tells you how much you're fluctuating up and down, which is a risk actually for hypoglycemia. And the consensus guidelines are that you should have levels less than 36. These patients are having values of 20%. So they do remarkably well. And we have CGM data to support that. So those patients that do well, not only do well, they do remarkably well. And this is data up to 11 years after a transplant. Not only that, what about survival? What about this concern about exposure of immunosuppression in the long run? So are you giving up the risk of increased mortality because you're exposed to drugs and immunosuppression and anti-rejection? So on one end, you have excellent glucose control, but maybe your life is affected, your quality of life and your survival because of the immunosuppression long-term. And we have been able to demonstrate that that's not true. That doesn't seem to be the case, at least not with our data. In our patients here, when you look at the cumulative survival, so if this starts going down, is patients dying. And at 20 years following transplantation, you have more than 90% survival of those patients, despite being on immunosuppression for those 20 years. And when we plot this information and this data with other studies that have looked at mortality risk in patients with type 1 diabetes, clearly these are not patients that have received a cellular therapy. These are patients that have been managed with state-of-the-art, with insulin, multiple daily injections, and so forth. We are down here in terms of mortality. So less mortality than other cohorts, and more than 90% survival after 20 years. Granted, the diabetes management is having a huge impact, but it's not being offset by negative effects of being on immunosuppression. So now let's shift gears. Let's talk about the site, location. Historically, we use the liver. Where do we get the islets? They come from a donor pancreas. We isolate them through the well-known Ricordi chamber and Dr. Ricordi's method of isolation. We get the islets and they go into the liver through a percutaneous approach through the alveology, and the islets flow into the portal system. They go into a vessel, they flow in there, and they lodge in the liver. But another important thing that I wanted to highlight also is that we are able to actually see the islets and that differentiate them and distinguish them for the rest of the tissue that we don't want. We want actually the islets when we're isolating them from the pancreas. This was a contribution from our own Dr. Alejandro. He's the director of the clinical cell transplant program. Before this stain that he developed, we didn't know what we were getting. I mean, we would isolate the cells, but we couldn't distinguish islets and the beta cells from the rest of the tissue. And it's because of this seminal contribution here at the DRI that everybody's using this in other centers when they isolate islets, because now you can distinguish them and know what is an islet and what is acinar tissue that is not endocrine tissue. And the islets again go into the liver, but what is the problem with the liver ultimately? It does work, it has been successful, but there are several limitations. Unfortunately, you put the cells within a vessel. So you can imagine that you're restricted by the volume. You cannot put too much volume within a vessel. You're going to injure things. On top of that, if you put islets in a vessel, you trigger clotting. Many of these islets can die because you form clots around the islets. So you need to anticoagulate patients. And you're puncturing a vessel to put the islets in. You have the risk of bleeding. So there are several limitations that make the liver not ideal. So we've been looking at other potential sites that could work. And one of those sites is an extravascular site. So we don't go into a vessel. and it's in the abdomen, in the omentum. The omentum is this fat tissue that covers the abdominal organs. And using what we know as a biologic scaffold, using plasma of the patient and thrombin, we generate this kind of like, it's like a little clot essentially that we form. So the islets are dripped down on the omentum. They form these little kind of like pancakes that they just attach on the surface of the omentum. We put thrombin, which activates the formation of a gel clot, and then the omentum is shifted with the hopes that those cells in the omentum will engraft so that they will get their own vascularity and ultimately release insulin into the bloodstream. And we were the first ones to demonstrate that this site, that other than the liver, actually worked. And we had a patient, and this is data, again, very similar to the one that I had shown you previously, where you have CGM data, the gray area here, limiting the values between 70 and 180. And you can see all of the values are in that range. This is up to 1 year data on the first patient that was transplanted. And this is just for to have an understanding of how variable the values, the finger stick sugars were before transplant and then after transplant. And most recently, one of those patients in the omentum has reached almost 5 years of transplantation. And these are the CGM metrics. An average blood glucose of 116. Look at the time in range, 94. Time below 70 milligrams per deciliter, 3%. And the recommended target is actually less than 4, and nothing less than 54. And this patient is on average on 3.4 units per day. So again, showing that this site can actually allow for engraftment of the islets. Clearly, this needs to be optimized. And we're really hopeful that with the funding that has been provided and it still comes in from donors, we can continue to explore this site and optimize it, because again, it does work. And we have evidence, at least in this patient, up to 5 years after transplantation with minimal insulin requirements. And we have outstanding CGM metrics. Shifting gears now, what about the source? And I'll talk a little bit about the stem cell derived islets. So when we talk about beta cell supply, strategies aimed at addressing the current beta cell supply, which is dependent at this point. What do we depend on? On cadaveric organ donors, right? These cells, we obtain them from a deceased donor, and clearly that is not enough to supply all the patients with diabetes, with type 1 diabetes that may benefit from such treatment. So the field has been looking at the potential of xenotransplantation with pig islets, modified pig islets, and most recently with the development of stem cell derived pancreatic islets, which I'm going to be talking about in a second. So a group in Harvard led by Dr. Melton was able, with this stepwise approach with 6 different steps, take embryonic stem cells and was able to grow them or differentiate them into insulin-producing cells, fully functioning islets. And this opened a whole new, window of opportunity and doors of treatment, because this can be used for in vitro studies. Now you have this source that you can do on demand, and you can actually assess them in vitro. We can transplant them to treat patients with diabetes, and you potentially can genetically manipulate them to provide kind of like stealth from the immune system, so the immune system doesn't recognize them, and hopefully being able to transplant this without the need for anti-rejection drugs. And with this advancement, with the advent of this islet derived from embryonic stem cells, this company Vertex was actually able to develop a clinical trial that was approved by the FDA, and we are one of the centers that was approved to actually carry out this study. But still requiring immunosuppression, because these are islets that are coming from a different human being, not the one, they don't share the same information as the recipient. And you may be aware with this report in the New York Times of the first patient transplanted under this protocol that has done remarkably well. And this is just to show that he was making his own insulin from these cells that were transplanted. And look at what happened. This is at 90 days after the transplantation of this product. Insulin requirements went from 34 to 3 units per day, A1C from 8.6 to 7.2%. And the patient was doing remarkably well, which is extremely exciting. And we are a part of this protocol and participating. And we really hope that this will change the field if indeed data like this are validated and we can replicate them in more patients. So now shifting gear to prevention and of diabetes. So I'm not going to show you all the data in prevention trials, and there will be too much to cover, but I want to show you data on a trial that I participated in with the DRI with TrialNet collaboration, and that actually showed efficacy. So you have to understand that prior to this trial, we had nothing that would actually prevent the onset of diabetes. Once you have those two antibodies, once the sugar starts to go up, you are essentially predestined to develop diabetes. And the risk is about 50% in the next 2 years, which is very difficult for a family to let them know, look, your child or you are going to develop diabetes, and you have a 50% chance in the next 2 years to develop type 1 diabetes. So this therapy, and I want to guide you to the slide to understand this better. The drug is called Plysma, that's the agent. And you have here proportion free of type 1 diabetes. So as the line drops, means patients getting diabetes. And the blue line is those patients that get the drug, and the red line is those that do not get the drug. And to understand if a clinical trial is actually meaningful, your data, you need to make sure that the group that is the placebo group is behaving as you would expect it to behave. I told you that if you have 2 or more antibodies, and if you have impaired glucoses, so you don't have diabetes, but your glucoses are not fine, 50% of those patients will get diabetes by 2 years. So look at what happens to the red line. At 2 years, 24 months here, indeed, 50% of those patients had diabetes. So you know that you started with the right patient group. They are developing at 50% rate type 2 diabetes. But the ones that got the treatment, look at what happens. Even at 6 months, you start seeing a difference. And these patients they slow down, and you have a reduction in the progression to type 2 diabetes. And the same investigators did an extension study up to actually 6 years, and that benefit is sustained. So finally, there is a therapy that can delay the diagnosis of diabetes in those that are at high risk for developing diabetes by about 2 years. In the extension study, they actually show that it can delay by 2.7 years. And the effects were greatest in the first 3 years after treatment. But what else have we learned that is critical to understand for the people that are following all the research in type 1 diabetes and that are providing with the funding that is critical to conduct clinical trials, is that one drug is not going to do the trick. And we're going to look at precision medicine and how a heterogeneous these diseases. We were able to learn that the ones that were most likely to respond with this therapy are the ones that have a particular genetic profile, the ones that have absence of this genetic marker, HLA-DR3. They have to have presence of HLA-DR4 and absence of these autoantibodies. So again, precision medicine becomes critical. We cannot look at this from the perspective that one drug will fix everybody. And we will have different therapies that tackle the immune system in different ways. And one therapy may work for one patient, but it may not work for the other one, and so forth. So I think all of these trials are helping us understand and get that concept of most likely you need multiple agents, and different patients will need different agents. So let's go even earlier in the disease process. I'm sorry, a little bit later in the disease. So I showed you prevention. Now let's look at new onset trials. So we have learned that many things do not work, but are there some interventions that may actually slow down? Remember, these are patients now that have the disease, have been diagnosed, and you want to try to rescue whatever amount of C-peptide insulin they're making and slow down the process of loss. And this study that I was also able to participate to TrialNet tested a medication called thymoglobulin, which is actually the standard immunosuppression treatment for islet transplantation. And we're using it, we tested it in new onset patients, and they combine it with another medication called GCSF, which is a drug that can release and mobilize white blood cells from the bone marrow. And the idea that was that this could actually favor cells from the immune system that can abate and slow down the immune activity that is happening, the autoimmunity early on in the disease process. So some patients got only the thymoglobulin, here as shown as ATG. Some patients got the thymoglobulin plus this G-CSF, and there was your placebo, your control group. It was 89 subjects.
A turning point in the war, 3-month war, and Ukraine's top military commander tonight saying, and I quote, "Tonight we're not just defending ourselves."
I have a little bit of background in there. And you can see that that the placebo group, the one that didn't get treatment, starts to slow down, and you have the loss of insulin production, and it comes down and it continues. This is the slope. This is what we expect to happen. But you see that the ones that get the thymoglobulin with this G-CSF, there is a dissect, so it opens up, right? The lines separate, but these were not statistically significant. And in the ones that got thymoglobulin only, the difference between the blue line and the red line is actually significant. So you do have a delay in the in the loss of C-peptide with this treatment. Is it perfect? No. Is it clinically meaningful and relevant for patients? Absolutely. The more that you maintain your own insulin production, the better it will be for your glucose control and the prevention of complications associated with diabetes. So now there's a therapy that, although not perfect, is relatively safe. There were a lot of children included in this study. It was only 2 injections that were given, and the patients did well, and you slow down the progression of loss of C-peptide. And now let me actually discuss a little bit about the POSEIDON trial, which has the same kind of rationale. We want to intervene early by giving medications that can modulate the immune system and inflammation. These are safe drugs. These are actually safe agents that you can buy over the counter, omega-3 fatty acids and vitamin D, with the hope of maintaining or preventing loss of C-peptide in patients that have recently been diagnosed, and even in patients that have had diabetes for up to 10 years, if they still have some insulin production, because it's expected that they will still have some inflammation of and autoimmunity, and maybe we can rescue those cells, those insulin-producing cells. So again, this study was designed to test the safety and the efficacy of high doses of omega-3 fatty acids and vitamin D. We divide the patients. We actually have adults and children, and we divide the groups based on whether they're new onset disease up to 6 months, or if they have established disease beyond those 6 months. And they get randomized to either getting omega-3 fatty acids combined with vitamin D or vitamin D alone. And then they get treated for 1 year, and we follow them for another year after stopping the treatment. These are the agents that we've been using, a very highly purified form of omega-3 fatty acids. A lot of the omega-3 fatty acids out there are not highly purified, so we have control on the purity by using this product, and also we have a manufacturer that we can also make sure of the reliability and the consistency of the product for this clinical trial. That's for the vitamin D. The major inclusion criteria are the same as any new onset trial in type 1 diabetes. They need to have diabetes because of the autoimmunity. We need to check for antibodies, and these patients should have C-peptide, insulin production, because if they have no C-peptide, what are we rescuing? So we need to have some beta cells so that we can rescue them. It has been a challenge, unfortunately, with this study. We started with high hopes to recruit and finalize this study early on, and then we unfortunately encountered challenges, which had been challenges in other clinical trials. It's typically much easier to enroll children who are much more motivated in the new onset space than some of the adults, unfortunately. Time off from work and school has been one of the major limiting factors. And then the COVID-19 really halted everything, and it severely affected this study, unfortunately. We have been left at, from a total target of 56 patients, it was going to be 28 children and 28 adults. We've been able to randomize 23, so almost half of what we expected to have done. Mostly children, as expected. 13 subjects completed 1 year of treatment. We have 12 children and 1 adult. And we have learned that the tolerability to these high doses of vitamin D and fish oil has been excellent. And we have had no study-related serious adverse events reported to date. So we are excited to continue with this study because we want to have an answer, and hopefully, we will be able to continue. There's a little bit of an uptake at this point with the pandemic slowing down. So hopefully we will be able to continue recruit patients and finally have an answer on whether this intervention has an impact in diabetes. And I showed you earlier the data on that drug called the Plysma that successfully delayed the onset of diabetes by about 2 0.7 years compared to patients that that were treated on placebo. And that same drug now is being tested in new onset patients. It just makes sense, right? If it has an effect early on in the disease to prevent it, can it have an effect if you treat new onset patients to slow down the loss of C-peptide? So I've been participating in this trial, and I'm excited to be a part of it, and hopefully we'll learn from it in the next few years. So in summary, I've taken you a little bit throughout the history of what we've done with islet transplantation and how our institute has moved the field in that arena, how we collaborate beyond just the cellular therapy as a cure in other stages of the disease to try to tackle type 1 diabetes. Some of the exciting trials that I'm currently participating in, and some other trials that will be coming along, promoted by Dr. Pugliese and Dr. Schuyler, that I'm also very excited that will be starting soon. And I cannot continue without acknowledging, the huge group of people and that I'm blessed, I'm fortunate to work with, medical people, administrative, regulatory, the TrialNet team, and the huge amount of physicians and minds going from basic scientists, clinical scientists, clinicians, educators, immunologists that play a critical role for all that we do in our efforts to cure this disease. And clearly, we have all the funding and other agencies that have also helped us and believed in our mission. And with that, I'll end, and I'll be happy to take questions.
That was great, Dr. Baidal. Thank you so much. I have a question as it relates to the Vertex trial, because I know it's one that we get quite often, and we know we saw great success with the first patient, and then we now understand that there is a pause on the trial itself. Can you talk a little bit about that and kind of where we stand and what kind of next steps are with the Vertex trial?
Yeah, absolutely. You can imagine that we're a little bit surprised as is the company, as to what are the details on the hold, on the clinical hold. And what we know is that it appears the FDA feels that there is not enough data at this point to justify, and let me give a little bit more details about this. This trial is a first-in-human study, and it was testing a particular dose of those embryonic stem cells. And the first couple of patients were to be dosed with a dose of cell product that was felt to be safe and hopefully will have an impact. But ultimately, the idea was to provide a larger dose of cells with the hope and the target of rendering patients insulin independent with this larger dose. Based on the data safety, the safety data on the first two patients transplanted, then there was this interest in moving forward with the trial to provide a higher dose. But the FDA has said, wait a minute, we don't have enough data that justifies increasing the dose. So that's all we know. What additional data the FDA wants? Do they want just to wait a little bit longer follow-up on the two patients that have been transplanted? We do not know. So that's discussion that is going with the sponsor and the FDA. I feel this is something that will be temporary. As long as Vertex and the patients do fine, and it's demonstrated that there is efficacy and safety. Maybe they just want longer data to feel comfortable with it. Again, and I kind of understand the FDA's input here. Safety is critical whenever you have a new first-in-human trial. But I think my bias is that it's a temporary thing. The patients, I'm hoping, will continue to do fine. So far, there has been no red flags from the perspective of any serious related adverse event to the product, which is the first thing that you look on in these studies. So in the absence of that, safety is maintained, I think the FDA will likely lift this. But it remains to be seen, and my information is, again, limited to what is posted on the Vertex website.
Great. Thanks. Any other questions for I have a bunch of questions. Any other questions? Laura.
Dr.
go ahead.
Baidal, oh, go ahead, Laura.
Sure, I'm going to cut you off. I'd like to ask you a question as an endocrinologist. I mean, this all sounds so exciting and hopeful, but I want to know, what about now? Is there a mechanical cure out there now? Are these insulin pumps and CGMs working in a closed loop system that will help people maintain normal glucose levels without needing their inputs? Because it's like, I mean, for 20 years, honestly, I don't mean to be rude, 20 years we've been saying it's right around the corner. And I would like to tell my daughter, now you can go on this system and take you out of it, which would really change her life like you couldn't imagine.
Absolutely. I think, so let me put on my clinician hat, because that's what I do all the time in the clinic. So I feel that there's a couple of things that I need to make the point clear. I think the technology we currently have is unique. If you look at it, and also it is faster for technology to advance in type 1 diabetes, because you have a huge amount of patient population and is relatively risk-free with what we currently can provide, right? So those are trials that can be done quite quickly, and you don't need years of data to demonstrate efficacy. So that's why a cellular therapy that has to go to all the rigorous FDA and clinical trials and years of data takes longer, as differs to something that in the clinical aspect with technology, clearly technology can move faster. And to me, also putting back my scientist hat on, I think technology allows for a bridge before we can have that biological cure. So it's a great option, and absolutely every single patient, the way that I see it, every single patient with type 1 diabetes right now should be offered and should be made, and should have accessibility to current newer technology. It's a standard of care. I mean, whoever is not already on these technologies is a step backwards. So going to your question in terms of a closed loop system, right? We can talk about this, but I'll try to make it brief. What we currently have is hybrid closed loop systems. It's a hybrid because what the system does is automation of insulin delivery, taking care of your basal insulin needs. Those devices are very effective at taking care of your basal insulin requirements. And they're very effective during the night, when you're not eating, when you're at rest, when you're not exercising, you're sleeping. The algorithms that have been developed are very good at keeping the blood sugars very tight. The issue is prandial requirement. Every time you eat, if you need a correction. So you can imagine the development of an algorithm that is able to identify that you're eating, so without you doing anything, give you insulin that is going to be quick enough to resemble what happens in your pancreas, where the insulin goes directly into the venous system. This is still subcutaneous, right? It needs to get absorbed under the skin, get into the bloodstream, right? All of that is a huge challenge. Nonetheless, actually, in the next couple of weeks, there is at the ADA, there will be a presentation by a group in Harvard actually testing a device that is having an algorithm where you do not need to provide any input. The system will learn and is supposed to provide changes and insulin administration independent of the user. So that data remains to be seen. I'm excited because the field is moving great. And if you're able to eliminate user input, and it shows efficacy, right? Maintaining sugars in the postprandial state, well, in good ranges without user input, will be unique. And that's where this technology is going, and that will close the loop, right? You let the algorithm take over. Are we there yet? No. Are we close to that algorithm? I think we're getting there. And there's still the question of, do you need insulin only? Do you need glucagon and insulin? Do you need another hormone to maintain that? Diabetes is not insulin only. You have deficiency, of insulin, you have too much glucagon, and you need glucagon to prevent hypoglycemia. So getting back to your question, I think your daughter should be on, at a minimum, a pump, and
Yes.
hopefully an automation of insulin delivery system now. And that field is moving forward. But again, that will never, unfortunately, be a cure for the disease.
No, I
It's a
know.
patch that it works very well, but it's going to be a bridge that provide, and it's going to be accessible to a multitude of patients that can benefit now from it until we develop a cure. But yes, hopefully your daughter will be in those technologies.
Right. Thank you.
Amy, I think you had a question.
I did. Dr. Baidal, for the cohort that you're studying with the 20-year group, what milestones do you think you'll look for in the next 10 years beyond, time and the things that you're already studying? Or, I mean, if you were to, their data so well, what are you thinking you'll see, or what are you thinking you hope to see as time goes by and that population ages a little bit?
Right, so there's two things. One is a 20-year survival, right? And then we do have data up to 10 years with insulin independence. One of the issues, right, is that keeping engagement. And remember, these are patients that were transplanted years ago, right? And trying to maintain them engaged, first of all, is an issue. We need the funding, bringing them. So I want to also highlight the funding allows for us to continue monitoring those patients, because the question that you pose is a critical question. So first of all, again, engagement, try to keep them, flying them in, because many of these patients are out of state, and try to see where are they. So what are the critical questions in those patients, in those that still maintain function and they're still making insulin and are on immunosuppression? First of all, is it still safe? That's the first thing, right? Is it still safe? They may be living the same, but are they having a lot of infections or problems? What's happening with the immunosuppression, right? So first of all, safety. Second is, which is one of my biases, I think that after many years, you may reach a status of engraftment and immune equilibrium where you're fine. You're likely not to lose anymore. Probably everything has quieted down and you develop some sort of tolerance now. Can we start getting them off a little bit of the immunosuppression many years later? Maybe I'm wrong. I mean, I don't have a way to, at this point, it's a hypothesis, right? But some patients do, so many years later, very well with very low doses of immunosuppression. And we should study that, but all of that requires, blood work and studies and immunological testing and so forth. But again, so first of all, safety. Second, are they still maintaining their own insulin production? Is it changing? Is it the same? Incidence of complications, diabetes-related complications. Are they having less eye disease, less kidney dysfunction, less neuropathy? Incidence of cardiovascular disease, which is what most of these people die of. Are they having less heart attacks? I mean, what's going on from that perspective? And quality of life. Has this changed their quality of life? 30 years down the road from when they were initially diagnosed, from where they were before they got the transplant, can they go back and say, this has been something that has changed my life and I will do it all over again? So those are the questions that I would like to address as I monitor them through the years.
So I have a question regarding the gaps in kind of the research and what in your opinion is needed for us to cross the finish line, because we hear, right, Vertex, and we hear ViaCyte, and we feel like we're kind of making progress in that realm, but what's missing right now in the research space to get us over that final line?
It's a very difficult question. I'll try to address it the best way that I can with my own bias. And I'll focus it on, again, the biological cure, right? So at that stage of the disease process. First, we need the product, right? So let's talk about the product, right? We have islets that come from a donor and the source is gonna be a problem. So having the stem cells, if this actually pans out, again, it's a first in human trial, we don't know. And one of the potential caveats with these cells is that can they become dysregulated? Can they start producing other hormones, escaping the normal function, because these are created in a lab? So those are things that need to be addressed. So first of all, we need to have a safe product that we know is fully functional. That's the first thing. Maybe Vertex has that product. I think it's too premature to jump into it and make conclusions. So that's the first thing, having the product, whether it's going to be continue to be pancreatic islets derived from donors, whether it's going to be mesenchymal stem cells, whether it's going to be from pig islets, whether it's going to be the same patient cells that can become regenerated. First, we need to have the product. Secondly, we need to create tolerance. It's critical. We need to create tolerance so that the body and the host is able to accept those cells and not reject them. And we are going to be actually testing a trial, as you know, with Dr. Kenyon, the anti-CD4 ligand, which is extremely exciting. There's a trial to come that will try to address that question. Can we modulate the immune system to develop tolerance? So if we tackle the product and the source, and we tackle the immunity, the rejection, by making the product escape immune recognition, we have solved a huge barrier. The one that remains is, where do we put them so that they don't die and that they stay there and that most of them survive? So that's the location. And so far, the question is, do you need an intravascular site? Is an extravascular site going to be better? And that remains to be seen. At this point, if you do an extravascular site, you need to make sure that that product gets quickly revascularized. It needs to get vessels very quick, because otherwise it's going to start dying and dying and dying until vessels are formed, right? Which is what we have learned from the Omentum, unfortunately. They do survive, but you're likely losing a lot because you're outside of a vessel. So that's a little bit the way I view it, right? So having an adequate product that works effectively and that you can use on demand, hopefully stem cells will be that product. Being able to make them escaping immune recognition, that would be the second issue, and lodge them and put them in a site where they will engraft, causing minimal problems to the subject, and where they will survive, hopefully, for the lifespan of the subject, although it may not be required that they survive for the lifespan of the subject. Potentially, you could re-treat the patient, right? You know, every year so often, as long as you have this source of cells, and they may be fine. But ideally, that's the way that I will envision this.
That's great, thank you. Any other questions? Great. Well, I want to thank Dr. Baidal for taking time to join us this evening, and certainly, thank him for all of the amazing work that he's doing in the lab and spending time with us this evening, explaining what he's doing in the lab. I always feel smarter after I come to one of the Meet the Scientist events. I hope you do as well. And I hope that you can see that the dollars that come into the foundation are really supporting what we find to be just the most amazing science as we advance towards a cure. And hopefully we'll get there sooner rather than later, and that will be thanks to everybody's help on this call. So thank you all so much for joining us tonight. If you have any other questions, don't hesitate to reach out to myself or anybody on the team, and we wish you all a safe evening, and thank you all.
Thank you.
Good night.